Hello everyone, my name is Patricia Melogr and I am a working clinical laboratory technician in the Transfusion Services Department of Duke Regional Hospital. We're a 370-bed hospital within the Duke University Health System in Durham, North Carolina. I'm here today to propose a study that compares how well D variants are detected using the two most common automated platforms for pre-transfusion testing. There have been many studies done comparing the efficacy and performance of antibody detection between gel column agglutination and solid phase technology. However, there is not much information comparing the two methods with forward typing. Particularly important today is the ability to detect D variants in each method. Currently, the two competing automated methodologies are gel column agglutination and solid phase technology. Most medium to large volume laboratories utilize one of these methods. Smaller laboratories may use manual gel column agglutination or tube testing. The Duke University Health System is unique that there are 3 hospitals that have transfusion services, and between us, there are 2 different automated methodologies for pre-transfusion being used. It has been observed that on occasion, our facility, which uses automatic gel column agglutination, will detect D variants that are not picked up using solid phase, causing a historical mismatch. The patient will have typed Rh negative at one facility using one method, but Rh positive at another facility using another method. All facilities use SafeTrace as their blood bank software. Therefore, patient history is visible to all hospitals no matter where the patient may be. It is suspected that gel column agglutination is a better method for the serological detection of D variants in routine pre-transfusion testing. The Rh blood group system contains 50 defined antigens. In clinical practice, the most significant antigens are D, big C, big E, little c, and little e. D is the most significant antigen because of its immunogenicity and its corresponding antibodies' devastating history. The frequency of the D antigen is 85, 95, and 99.5% in Caucasian, Sub-Saharan African, and Eastern Asian, respectively. I think we're all familiar with the mechanism of the maternal sensitization that leads to HDFN, so I'll only say this. While the number of deaths from HDFN have been greatly reduced by the advent of Rh immunoglobulin in the late 1950s, it still exists as a threat to Rh negative and D variant mothers. It is important to understand the genetics of Rh blood group systems, which is comprised of 3 genes, the RhD, RhCe, and RhAg genes. RhD and RhC are located on chromosome 1, but are in opposite orientation. RhD is arranged with 10 exons in the 5', 3' direction, while RhCe is arranged with 10 exons in the 3', 5' direction. The proteins coded by these genes span the red cell membranes 12 times, with the internal N and C termini and 6 potential external loops. Individuals who are D negative inherit 2 copies of a silent allele that does not code for proteins. Because the RhD and RhC genes are in close proximity, molecular changes can occur and cause variations of the proteins that the genes themselves code for. Changes affect antigen expression in many ways. Any change that affects the D antigen, whether it is the epitopes themselves or the number of antigen sites, are collectively termed D variants. There are 2 molecular mechanisms that cause D variants: amino acid substitutions in the RhD antigen caused by nucleotide change in the RhD gene, and genetic recombination, which results in gene conversion, where a section of the RhD gene is replaced by the corresponding section from the RhCe gene. In the European population, all known partial D phenotypes combined are less than 1%, and the gene conversion is typically a single missense mutation. However, in individuals of African descent, the occurrence of aberrant RhD alleles and the ability to form anti-D is much more frequent and is often caused by multiple missense mutations. The location of the amino acid sequence change is important to note. For weak D, these changes occur within the cell membrane or intracellularly. In partial D, the change occurs on the outside of the cell membrane and are therefore externally exposed. In Caucasian populations, about 15% of women serotype as RhD negative and are homozygous for the D gene deletion, most frequently on the r haplotype. For African populations, on the other hand, around 67% of D negative individuals carry at least 1 copy of an inactive pseudogene called the RhPsi, and 15% carry a non-functioning hybrid gene. D variants are often put into 3 categories. First, there is a D mosaic, a qualitative variant which has been determined to lack certain epitopes of the normal intact D antigen. Individuals with this variant are susceptible to making alloantibodies to the portion of the D that they are lacking. A better term for these individuals is partial D, but the best term is D variant. Partial D variants can be put in Roman numeral categories D2 to D7. The most common D variant in Caucasians who have made anti-D is the D6 variant. In African Americans, it's the D3A and DAR. Weak D, which is previously known as DU, is a quantitative variant defined as having normal intact D antigen with fewer antigen sites, thus making D expression weaker. There are many categories of weak D, but types 1 through 3 account for 93% of weak D variants within Caucasians. Weak D, 1 and 2 have fully intact D antigens. Based on cases of individuals with these weak D variants, evidence suggests that it may be unlikely for them to make anti-D when exposed to D-positive blood. However, other weak D variants are known to make alloantibodies when exposed to D-positive blood. This is part of the reason that it is not ideal to use the weak D/partial D terminology. It is best to use the term D variant for both. DEL is a rare D variant with a very weak representation of D antigen sites. These individuals cannot be detected with conventional serological methods, so I'm not going to go into much detail about this phenotype now, as they will remain in the serological D negative category. The only way to know what type of variant an individual has is through molecular testing. Currently, in many parts of the world, IAT D testing is performed on blood donors and newborns from D negative mothers. It is logical to want to ensure that the blood not be falsely typed as D negative when the donor is actually D variant. However, it seems feasible to perform molecular testing on D negative mothers, because if they do turn out to be weak D type 1 through 4, they will not be a candidate for RHIG, which will reduce their exposure to human blood products. They will also not need to receive Rh negative blood. It is also possible to molecularly type the fetus to see if the mom is an Rh candidate or is at risk for HDFN. So a comparison between the two leading automated serological methodologies used in most routine immunohematology labs will be conducted to compare outcomes for D variant individuals. Comparison of both platforms will allow conclusions to be drawn about the sensitivity of each method. In order to obtain an adequate pool of specimens, outreach will be made to request aberrant, discrepant, or questionable results for RHD typing from immunohematology laboratories nationwide. Informed consent will be obtained from participants by the laboratory in which they request for further study is made. Laboratories are instructed to remove or conceal all patient identifiers prior to mailing the sample and will be provided with a prepaid shipping label. On arrival, samples will be given new identification numbers. The two automated instruments to be used are the Vision, manufactured by Ortho Clinical Diagnostics, and the Echo, manufactured by Immucor. A third method, Immucor's Precise Type HEA bead chip kit, will be used to validate the serological results. The principle of the Vision is gel column agglutination. A gel matrix sits in a fluid containing known anisera in a narrow well on the plastic card. A suspension of red cells is made and pipetted onto the surface of the gel. If antigen is present, antibodies in the anisera will attach to the corresponding antigen and agglutinate the red blood cells. Gel cards are then centrifuged and agglutinated cells will be caught in the gel matrix at some point. Unagglutinated cells will not be impeded and will be allowed to freely centrifuge to the bottom of the well, appearing as a thin cell button. The Echo uses solid phase hemagglutination to determine blood type. This is a technique in which one of the components of the antigen-antibody reaction is immobilized onto a solid medium, and after reaction with free antigen antibody, the endpoint of the reaction is indicated by the use of red cells, which may be part of the antigen-antibody reaction or may be added as indicator cells. In forward grouping, U-shaped microplate wells are coated with anti-A antiserum and anti-B antiserum and anti-D antiserum. If antigen-antibody reactions occur, they are spread across the well to varying degrees in a diffuse layer after centrifugation. If antibody-antigen reactions do not occur, a bold cell button will be visible at the center of the well. Other differences between these two methods must be taken into account. This study is concerned about RHD typing in the D variant individual, and therefore the clone used to produce the anti-D within the test system should be noted. Anti-D used in gel column agglutination is derived from a single cell line, MS201. Anti-D used in Immucor's solid phase hemagglutination is a monoclonal blend from two different heterohybridomas. Specimens will be run in duplicate on the Vision and the Echo. The results will be documented. Specimens will then be placed in a refrigerated storage until 20 samples can be collected. After 20 specimens are obtained, they will be batched and prepared for Precise Type HEA bead chip molecular assay. The Precise Type HEA bead chip kit is an in vitro diagnostic test intended for the molecular determination of allelic variants and that predict erythrocyte antigen phenotypes. The Precise Type bead chip test uses the proprietary elongation-mediated multiplex analysis of polymorphisms, or EMAP, technology to identify the presence or absence of selected alleles associated with a given phenotype. Once allelic variations have been molecularly determined, samples will be grouped by type of devariant. Each group of devariant will be compared to the results of the vision and the echo to determine how many samples were properly identified as devariant and how many were missed, in an effort to determine which method is better capable of detecting devariance. It is essential to properly type certain populations for RHD. The type of RHD reagent that is used is important, but what role does the method of detection play? A focus within the routine immunohematology laboratory, in light of a shrinking donor base, is the need to preserve Rh-negative blood products for those who truly need it. Falsely typing an individual as Rh-negative when they are truly a devariant with no ability to become immunized, for example weeks D type 1 through 4, is one way to unnecessarily waste Rh-negative products. Falsely typing an expectant mother can also have consequences, such as exposing to her to Rh-IG prophylaxis, which is a human blood product and carries the same risks as other human blood products. Other risks like fever, chills, and pain at the injection site, mild and severe intravascular hemolytic reactions. It is suspected that gel-column agglutination is a better method for serological detection of devariants in routine pre-transfusion testing. A side-by-side comparison between the two dominant automated methods will give insight into which method might be more suitable. Until molecular genotyping is broadly and affordably available to all immunohematology laboratories, it is useful to know which method performs better, especially if the laboratory is located in an area with a large Rh-negative or African American population. This comparison study will also shed light onto which variants will not be caught by either method. Thank you.